Hello everyone, Professor Big here. Today we're going to talk about the cellular blueprint. We talked about the overview of pathophysiology in the previous lecture. Now we're going to focus on the cell itself. Now along with this lecture, please look at your key points in your module, and you can use your study guide to take notes as we go through this. So, we often view disease through large-scale symptoms, but clinical failure actually begins at the microscopic level. Today we're going to look at the diagnostic breakdown of how cellular adaptation and injury ultimately lead to systemic disease in the population. So, to understand a sick patient, we first have to distinguish between the three fields. Physiology is the study of the healthy body, the normal operational parameters. Pathophysiology is the mechanics of the disease, right? The what of the disease. It focuses on the step-by-step sequence of events at the cellular level when those standard functions fail. Finally, you have pathology, which is the anatomical identification of disease through physical alterations and changes, such as those seen in biopsies or autopsies. Fundamentally, disease is homeostasis gone wrong. The body maintains health through the negative feedback loops, which act like a thermostat, which corrects deficits or excesses and sets it back to the set point. When stressors overwhelm these loops, a disease process begins, right? This timeline follows a specific chronology. It starts with the latent or incubation period, which is called the silent phase. Then it moves to the prodromal phase, when that's the stage when people have vague or non-specific signs like fatigue, for example. Then it reaches the acute phase, which is the peak manifestation of the disease, and ends with convalescence period, which is return to the normal state. Think of the cell as a factory, right, with specific vulnerabilities. The plasma membrane is the security gate. If it loses integrity or doesn't work, harmful agents can flood the cell. They can get in. The mitochondria are the power plants. If they fail instantly, actually, during oxygen deprivation, and that is called hypoxia. Lysosomes handle waste, but their rupture causes autolysis, where the cell basically digests itself. Finally, defects in the ribosomes or the ER lead to protein synthesis failure. This causes cell starvation. The sodium potassium, I tend to say potassium instead of potassium sometimes. The sodium-potassium pump is the most important engine in the cell. It constantly uses ATP to push 3 sodium ions out for every 2 potassium ions it brings inside the cell. Why does this matter? Because sodium naturally draws water. By pumping sodium out, the cell prevents itself from overhydrating. If ATP production drops, the pump fails. Sodium falls back in, water follows, and the cell suffers fatal swelling because water follows salt. Now going back to etiology. Etiology refers to the cause of disease, as we mentioned in the previous lecture. These agents can be biological pathogens, chemicals, toxins, like for example toxins or high blood glucose. It can be physical, things like trauma or extreme temperatures, or deficits like nutritional starvation. We also use specific clinical terms, such as idiopathic, means the cause is unknown, while iatrogenic refers to a disease caused by medical treatment or procedures, such as bladder infection from an indwelling catheter. This is a list of causes for cellular damage, for example, ischemia, physical agents, mechanical damage, chemical toxins or foreign objects, pathogens, abnormal metabolites, nutritional deficits, fluid or electrolyte imbalances. All of these can cause cellular damage. When the cell is stressed, it tries to adapt, right? Atrophy is the reversion to a smaller size. So basically, all of these are adaptation of the cell to stress. So atrophy, reversion to a smaller size, like muscle shrinkage during paralysis, where people develop muscle atrophy. Hypertrophy is an increase in the individual cell size, often seen in heart cells due to hypertension. When a patient has hypertension and it's chronic, basically the heart cells tend to go through hypertrophy. They enlarge. Hyperplasia is an increase in the number of cells. More concerning are metaplasia, replacing one cell type with another. Then you have dysplasia, when you have deranged abnormal cell growth. Neoplasia represents uncoordinated autonomous new growth, which we recognize as tumor. All of these are ways in which the cell adapts to stresses. This is a visual comparison which highlights the structural differences between normal tissue and adapted tissue. Notice how dysplasia shows disorderly growth compared to the uniform look of normal cells. Identifying these patterns is critical for early diagnosis, as some of these shifts are precursors to more serious conditions like cancer. It's important to distinguish between these two growth pathways. Hypertrophy is about size. Cells physically expand to meet demand, which can be physiological. For example, exercise, right? Your cells, go through hypertrophy to kind of meet the demands of the exercise that you're doing. Or pathological, for example, hypertension. Hyperplasia is about the number. It involves cellular division and can only occur in cells capable of mitosis. A common maladaptive example of hyperplasia is the formation of keloid scars. Metaplasia is a survival tactic of the cell. And for example, GERD, the flat squamous cell epithelial cells of the esophagus are constantly irritated by stomach acid, because remember, that's regurgitation of the acid into the esophagus. To survive, the body reprograms them into tall columnar stomach-like cells that provide a stronger barrier. However, there is a dangerous trade-off here, though. Prolonged irritation can push this metaplasia into dysplasia, which precedes cancer. So you don't want to keep that prolonged stress on the cell, even though it is adapting, but prolonged exposure to that stress can cause negative effects. So this is the domino effect of cell death. It begins with ischemia, diminished circulation, which forces a metabolic shift to anaerobic pathways. This causes lactic acid spike, dropping the intracellular pH. And we'll learn a lot more about pH later, but it's extremely important to maintain that pH. That is part of homeostasis. Consequently, the sodium-potassium pump fails, leading to fatal swelling as water floods the cell, eventually causing membrane rupture and death. The endothelium is the active tissue lining our vessels, all our vascular system. It is highly vulnerable to constant stress from hypertension, high blood glucose, and free radicals. This damage leads to two major clinical outcomes: atherosclerosis, localized plaque formation, or an aneurysm. This is where the vessel wall weakens and balloons outward, as seen in a cerebral aneurysm scan that you can see on the screen on the bottom right. When these metabolic processes are disrupted, substances accumulate within the cells, and this leads to dysfunction. For example, fatty liver occurs when hepatocytes accumulate lipid vesicles. Jaundice is the yellowing of the sclera due to bilirubin buildup. Finally, anthracosis or coal miner's lung. Sorry, I can never pronounce anthracosis. Happens when macrophages cannot digest the inhaled carbon particles, permanently blackening the lung tissue, as you can see on the screen. There are two ways a cell dies. Apoptosis is programmed, organized self-destruction. The cell digests itself into vesicles and does not trigger inflammation. This is normal. Necrosis, however, is the irreversible death from overwhelming injury. Now, that is not normal. In necrosis, the membrane disintegrates and lysosomes rupture, spilling harmful contents into the surrounding tissues. And as always, it initiates a systematic inflammatory reaction. To conclude this, lecture, remember that every devastating clinical diagnosis begins as a microscopic struggle for balance. Remember, homeostasis. It starts with an etiological stressor that causes the loss of homeostasis. And we did talk about the different types of stressors, that list of the causes for cell death. Then, when the homeostasis is lost, the cell attempts to adapt. But if the compensation, its adaptation fails, injury and necrosis will occur. Only then does it manifest as a systemic disease with recognizable clinical symptoms. And to treat the patient, we first have to understand the cell and how it adapts to injury. And that is it for this lesson. All done, says the happy cell.